Hello everyone, my name is Sumbul Akil and I am a final year PharmD student from University of Peshawar, Pakistan. I enrolled in this course because it seemed like an opportunity for choosing any specific career side, and it helped me a lot. I am expecting, well, firstly a certificate of accomplishment, and secondly a more confident myself out there in the field. The drug I selected is Docosanol, which is a licensed OTC topical antiviral that is for oral herpes medicine, and it got approved in July 2000 after its clinical trials by FDA. The drug is manufactured by Avenir Pharmaceuticals and available in markets of US and Canada by the name of Abreva. It is also available in European market by the name of Erazibam. The reason I choose this drug because it is the drug which is approved by FDA for only one clinical use, and if one goes through its monograph, the other might be uses are not even tried to be studied. So Dr. Etwati wanted a unique drug. In my perspective, it is. So these are some index terms or another names that are used in literature for Docosanol. These are behenyl alcohol, and Docosanol, 1-Docosanol, and this one is more extensively used, and the last one is Docosyl alcohol. The structure is also given. It is a simple molecule with no functional groups. That is why the researchers found its behavior to be much more predictable. Chemically, it is a 22-carbon fatty alcohol. So these are the two dosage forms that are available in the market for Docosanol. One is topical cream 10%, and the other one, which is new in the market, are the invisible cold sore patches. So I got it off from the company side, and that is why the consumer's perspective about the patches has yet to come. The inactive ingredients in the dosage form are light mineral oil, propylene glycol, water, sucrose distearate, and sucrose stearate. The reason that they are mentioned on the label of the drug is because any person could be allergic to anything, and not just active, it can be an inactive ingredient as well. So they come in 2-gram tube, 2-gram bottle pump, and the new one that I've already mentioned are the invisible patches. While I was collecting information about the dosage forms from internet, I read some consumers' viewpoints about this bottle pump, and they are not happy about it. They were complaining that there is a loss of drug and too much come out. So this needs to be taken into consideration. The general information that is so easily available on internet about the drug is that it is meant for recurrent oral facial herpes episodes, or more commonly known as cold sores or fever blisters. The active ingredient is N-Docosanol, also known as behenyl alcohol. It primarily has antiviral activity against many lipid enveloped viruses, including herpes simplex virus. It is marketed by SmithKline Beacon, and it is available without prescription as an OTC product. So these are some properties of N-Docosanol, and as you can see it on the screen, it is pure chemistry. The importance of all these points is that they can help predict the nature or fate of the drug in vivo and in vitro. Up till late 1970s, Docosanol was nothing but another chemical compound, and its uses such as lubricants or surfactants, etc. were being studied. It was in 1980s and late 1980s that its other uses were considered, and human uses were the prime focus of that studies. Here are some other computed properties of the drug. The most important of all is the molecular weight. The molecular weight, it is important because it determines the equivalency relationship of a drug with its derivatives. The most common example is of prednisolone and prednisone. Prednisolone is this much gram equivalent to prednisone. So this is how they do it with molecular weight. So a quick word about herpes simplex, because this is what docosanol is for. So this is a viral disease, and it is from herpes viridae family, and it is caused by both type 1 and type 2. The infections with the herpes are categorized on the basis of site of infection, that is, they are oral herpes if the visible symptoms are on the mouth or face, and this is the most common form of the infection. They are genital herpes. This is the second common form if the target is the genital organs. There are some other rare disorders also associated with herpes simplex, namely herpetic willow, cerebral herpes infection encephalitis, neonatal herpes, and possibly Bell's palsy. These are all caused by herpes simplex. So these are some images for oral herpes and genital herpes, courtesy of Google Images, just as Dr. Atwati suggested. Well, you can see it in the oral image that the site is unsightly, it is not beautiful. Maybe that is why docosanol got popular among the consumers, and maybe that is why FDA had to approve it with so minimum data behind it. So here's some more detail about herpes virus. It cycles between periods of active disease presented as blisters and between remission period. The active disease, it lasts for 2 to 21 days depending upon body's physiology and the immunity status. The genital herpes, however, remain asymptomatic. The cure for herpes, it has not been developed as yet, unfortunately. So that is why if a person is infected once, the virus remains in the body for lifetime, and there is always a risk of recurrent outbreaks whenever the body's immunity status is low. For example, in cases of HIV or cancer related immune suppression, or if the person is using some immunosuppressant medicine such as cyclosporine. In the later stages of the infection, or when the infection has aged, the outbreaks are often lesser in number, and in many cases the people don't experience them at all. So next in line is the epidemiology of herpes simplex. It includes all the three forms that I've mentioned earlier, that is oral, genital, and one of the rare forms. The worldwide rates for either type 1 or type 2 are between 60 to 95% in adults, with type 1 being more common than type 2. The rates of the infection are determined by the presence of antibodies against either viral species. In the US, 57.7% of the population is infected with type 1, it is more than half of the population, and 16.2% are infected with type 2. Among these 16.2%, only 18.9% are aware that they are diseased or they are infected. The rest doesn't even know that they are carrying the infection. During 2005 to 2008, the prevalence of type 2 was 39.2% in blacks and 20.9% in women. At this point, I would like to mention here that all the numbers I would come across on internet about the epidemiology of herpes simplex belong to developed countries, and there's a perception that the data from the underdeveloped countries is hugely manipulated. Well, this is wrong. The reason for underreporting from developing countries is the patient himself. People don't want to come in front or does not cooperate when approached. Next big problem is the follow-up. Developing countries lack monitoring and follow-up facilities, so it is not always manipulation. So in this slide I am going to narrate the society's perspective for the disease herpes simplex. It was not always stigmatized. It was merely a cold sore in an unusual place until the 1970s. As late as 1975, a study of physiological morbidity in a clinic for sexually transmitted disease does not mention herpes simplex because at that time there was no significant morbidity problem associated with the virus. During the R&D of acyclovir, marketed by Burroughs Wellcome, insisted that there were no markets for this compound. Most had hardly heard of genital herpes, thus marketing the medical condition separating the normal cold sore from the stigmatized genital infection was to become the key to market the drug, a process now known as disease mongering. Since the creation of the herpes hype, some people experience negative feelings related to the condition following diagnosis, in particular if they have acquired the genital form of the disease. Feelings can include depression, fear of rejection, fear of isolation, fear of being found out, and fear of self-destructive feelings. These feelings, however, lessens over time. Much of the hysteria and stigma surrounding herpes stems from a media campaign which began in late 1970s and peaked in 1980s. There were multiple articles worded in fear-mongering and anxiety-provoking terminology such as attacks or outbreaks or victims or sufferers. At one point, even herpetic word entered the popular lexicon. The articles were published by Reader's Digest, US News and Time magazine, among others. A made-for-TV movie was named Intimate Agony. The peak was created when the herpes, the new Scarlet Letter, entered the Time magazine in August 1982, forever stigmatizing the word in the people's mind. Herpes support groups have been formed in the United States and the UK, providing information about herpes and running message forums. People with herpes virus are often hesitant to divulge to other people, including friends and family, that they are infected. The advanced research about herpes simplex include a more precise targeted approach. There are two studies, one of which is from University of Florida. Researchers there have made a hammerhead ribozyme that targets and cleaves the messenger RNA of essential genes in HSV or type 1 herpes simplex virus. The hammerhead, which targets the messenger RNA of UL20 gene, greatly reduced the level of type 1 ocular infection in rabbits and reduced the viral yield in vivo too. The gene targeting approach uses a specially designed RNA enzyme to inhibit strains of herpes simplex virus. The enzyme disables a gene responsible for producing a protein involved in the maturation and release of viral particles in an infected cell. The technique appears to be effective in experiments with mice and rabbits, but further research is required before it can be attempted in people infected with herpes. The second study is from Duke University. The researchers are persuading this possibility to eradicate the HSV1 variant. So they are doing it by figuring out how to switch all copies of the virus in the host from latency to their active stage at the same time, rather than the way the virus copies normally, staggering their activity stage and leaving some dormant somewhere at all times. It is thought that the conventional antiviral drugs can kill the entire virus population completely, since they can no longer hide in the nerve cells. They developed one class of drugs which they are calling Entagomer. So this is chemically engineered oligonucleotides or short segments of RNA. So it can be made to mirror their target genetic material, namely herpes microRNAs. They could be engineered to attach and thus silence the microRNA, thus rendering the virus incapable of remaining latent in its host. Professor Cullen of the Duke University believes a drug could be developed to block the microRNA whose job it is to suppress the HSV into latency. So this was all about herpes simplex, and coming back to the original topic, which is N-Docosahexaenol, and starting off with its methods of manufacturing. It is produced by the reduction of behenic acid with lithium aluminium hydride as a catalyst. The formulations of preparation, the grade, it is technical, and in the market it is approved as 10% over-the-counter topical cream by FDA. It is being marketed by GlaxoSmithKline, as already mentioned. Docosahexaenol is listed as high production volume or HPV chemical in US, and chemicals listed as HPV were produced in or imported into the US in more than 1 million pounds in 1990 or 1994. However, these numbers have reduced over the years. The HPV list is based on the 1990 Inventory Update Rule, or IUR, the address of which is given in the bracket. In this slide, I have mentioned two laboratory methods or analytical extraction methods for N-Docosahexaenol. The first one is lanolin was saponified and extracted with petroleum ether. Neutral fraction of lanolin obtained was treated with digitonine, and cholesterol was obtained from digitonine positive fraction, while from the digitonine negative fraction several steroidal hydrocarbons were obtained, one of which was behenyl alcohol. Behenyl alcohol is the other name for N-Docosahexaenol. And the second one is 1-docosahexaenol was identified and determined in extracts of Pygeum africanum bark by gas chromatography. The links for both of these methods are given in the brackets. Establishing the PK parameters or pharmacokinetic parameters for a drug is the most important part in the drug development process. In this slide, I have mentioned the studies that led to the establishment of absorption, distribution and excretion statements for docosahexaenol. The absorption of docosahexaenol has been shown to be minimal under conditions reflecting normal clinical use. Of 209 plasma samples taken from 10 subjects 24 hours after multi-day test, only one had a docosahexaenol level above the quantitation limit, that is 19 nanograms per ml. In the other study, showing the distribution of the drug, long chain alcohols, which included docosahexaenol as well, were detected in developing rat brain at their highest level of 0.0109% of the total lipids at the age of 10 days and decreased to 0.0036% at the age of 40 days. The link for this is given in the bracket below. This is the experimental extract for establishing the elimination statement for docosahexaenol. In this experiment, a mixture of docosahexaenol and octadecenol were injected into the brains of 19 days old rats. The desired outcome was measured through radioactivity, and it was found that even after 12 hours of disappearance of octadecenol from the mixture substrate, docosanol was still there and was not used to any measurable extent. The link for this information is given in the bracket below. The three studies that I've mentioned in the previous slides led to the concluding points that bioavailability or systemic bioavailability of the N-docosanol, it is not appreciable. In another study using cadaver skin, it was indicated that dermal penetration of N-docosanol it is also limited, and it is confined only to dermis and stratum corneum, and does not even penetrate the deeper portions of the skin. So this is how they developed the dosage form, which is a topical cream because it carries all the necessary properties or desired properties, such as it should stick on, it shouldn't be oily, it should be easily wiped out, or etc. The metabolism or elimination data for N-docosanol, it is not fully determined as yet. The only thing that is available so far is that it is metabolized to N-docosanoic acid and incorporated into cellular lipids and phospholipids. This is the study abstract, which gave us the only information about the metabolism and metabolites of N-docosanol, which is N-docosanoic acid. The findings suggested that the anti-HSV activity of N-docosanol involves cellular uptake and metabolism of the drug. The link for the study or the address is given below. So this is the study abstract, which gave us the final agreed upon mechanism of action for N-docosanol. Before this, previous studies of the mechanism of action demonstrated that N-docosanol inhibits an event prior to the expression of immediate early gene products but subsequent to HSV attachment. This study cleared it out. It said inhibition of fusion between the plasma membrane and the HSV envelope and the subsequent lack of replicative events may be the predominant mechanism for the anti-HSV activity of N-docosanol. So they established that N-docosanol reduces viral replication and activity by effectively inhibiting the fusion between the plasma membrane and the herpes simplex virus envelope. The link for this study is given at the end of the abstract. Here are some introductions of N-docosanol in relation to other drugs. First one is overbane to heart is inhibited by long chain fatty acids and alcohols, and docosanol is one of them. Next is the anti-HSV activity of acyclovir was synergistically enhanced by the treatment of cells with docosanol as judged by the inhibition of progeny virus production and plaque formation. Let's move ahead and talk a bit about docosanol dosage and administration. It is so easily available on the internet everywhere. Like I got it off drugs.com, which is more reliable source in my opinion. So this drug is to be topically administered to the affected areas of the lips and surrounding skin as a 10% cream. It is not to be applied in or near the eyes or the mucous membranes, such as inside mouth or inside nose. It is important that the infectee must wash hands before and after the application of the product. For best results, remove any cosmetics prior to the application or reapplication of the cream. The cosmetics can be applied afterwards when the cream dries off. The infectee is fully capable of spreading this disease to another healthy person through this cream. Like if another person uses this leftover cream, he might get the germs of HSV. So that is why it is advisable that the infectee must use a separate applicator such as cotton swab to prevent the chances of spread. The pediatric dose of docosanol in children above 12 years of age is to apply 5 times daily until healed, but maximum use is for 10 days only. Use sufficient quantity to adequately cover lesions and symptomatic areas such as the areas with tingling for best results. It is important to use or start the docosanol at the early signs and symptoms such as tingling, phrenitis, redness and presence of a bump, etc. The adult dose for docosanol is same as the pediatric dose. It is to be administered 5 times daily and maximum 10 days. Maximum 10 days is the prescribing limit of endocosanol, and even if the problem or the lesions persists after 10 days, then you have to check it with your doctor, because it means that along with herpes simplex you are suffering from a secondary infection also. At this time we have no special population recommendations for docosanol. Special population means in renal patients, in hepatic failure patient, pregnancy data, in breastfeeding data, etc. These are some advices that the clinician or pharmacist or physician or any other healthcare provider are bound to entertain the patients with. The importance of docosanol usage at the early signs would be for best results. The therapy is to be discontinued after 10 days because if the lesions persist, it means you have a secondary infection also. Docosanol cream sharing, it is strictly prohibited because you might be spreading the infection. The docosanol cream is to be kept out of the reach of children and contacting the poison control center immediately if the drug is swallowed. The docosanol cream, it is for external use only, and this point is important, that if a dose is missed, apply as soon as it is remembered. Apply the next dose on schedule. If a dose is accidentally wiped off while blowing the nose or wiping the mouth, reapply the dose as soon as possible. It is important for the women to inform the clinicians if they are planning to get pregnant or they are planning to breastfeed their children. Why? Because we don't have any sufficient data at this time to provide its safety in pregnancy or in breastfeeding. Antidote and emergency treatment for endocosanol. It has already been established through pharmacokinetic studies that docosanol has poor absorption both orally and transcutaneously. Therefore, the potential for an adverse reaction related to an overdose is extremely low, and if something should happen, it would be purely because of a person's physiology and may vary from person to person. That is why the treatment is essentially symptomatic and supportive in case of any emergency. And the second para, I have listed some basic treatments in case of emergency, such as establish a patient airways or suction if necessary, or anticipate seizures and treat if necessary, flush eyes immediately with water or rinse mouth, and cover sunburns with dry sterile dressing after decontamination, etc. In the basic treatments, we studied ways and means which could be immediately applied to a patient and saving his life. However, in the advanced treatment, we study ways to restore the original health of the patient. That is why we consider drug therapies here, such as beta agonists, albuterol for severe bronchospasms or fluid resuscitations, or treating seizures with diazepam or lorazepam, etc. So these are some lines from different research papers and articles which could be traced back to even late 1970s, originally starting from 1975. In these research papers or studies, different researchers, they tried to link 1-docosanol with different environmental entities such as air, water and soil, and thus determining its environmental fate. I've already mentioned in this assignment that 1-docosanol was considered just a chemical compound up till early 80s, and its uses as lubricants or synthetic fibers or surfactants, etc. were considered. It was in late 1980s, originally 1986 to 1989, when its other uses were tried to be explored. This slide is also in context of the previous one. Here are some more studies which helped predict the environmental fate of 1-docosanol. These studies they established some pretty important facts about docosanol, such as the potential for bioconcentration in aquatic organisms is low. Hydrolysis is not expected to be an environmental fate process, since the compound it lacks functional groups that hydrolyzes under environmental conditions. These studies were not carried out to establish the efficacy and safety of 1-docosanol in human population. As I already mentioned, at that time docosanol was treated just like any other chemical substance. These studies, however, helped a lot once the researchers shifted to a whole new level of exploring 1-docosanol. Studies like these may be the reason that it got approved so sooner by the FDA, as everything about the drug was already established but in a different context. So let's talk about some non-human toxicity studies for N-docosanol. It is important to mention at this point that these studies were conducted post-approval or post-marketing of the drug. The importance of these studies or these experiments were to establish the efficacy of drugs in genital herpes and its effect on fertility and reproduction. So the first one was conducted on rats and rabbits. The behenyl alcohol demonstrated no effect on fertility or reproduction in rats when dosed up to even 1000 mg per kg body weight, and in rabbits with doses up to 2000 mg per kg body weight. The genotoxicity study was conducted, and it was found that behenyl alcohol did not increase the frequency of bone marrow polychromatic erythrocyte micronuclei in mice in vivo. The address for both of these abstracts are given with the respective abstract. In yet another toxicity studies for docosanol, it was found that it has no effect on weight and histological appearance of the prostate in intact rats, but it was found that docosanol increased adrenal steroid secretion in those rats. The other study that I've mentioned here, it is not post-marketing because it is 1977 study, but it does come under the category of non-human toxicity. It is a repetition, and I have already mentioned this study in distribution of docosanol. The link for the abstract is given at the bottom of the slide for convenience. So these are some values for the non-human toxicities, as you can see it on the screen. The lethal dose 50 for a mouse after 2 route administration, that is subcutaneous and intraperitoneal, it was found to be greater than 800 milligram per kg body weight, whereas in rats it is greater than 10,000 milligram per kg body weight after oral administration. These numbers and these values, they support the already established safety profile of docosanol, that it does not result in toxicity or overdosing after oral administration. Under safety and handling, we have storage conditions, which is directed towards the consumer or the user. It says store at or below 25 degrees C, that is 77 degree Fahrenheit, and it is to be protected from freezing. The second is disposable method, which is directed towards the manufacturer, and here it is taken as a chemical compound and not as a drug. That is why they have mentioned that the most favorable course of action is to use an alternative chemical product with less inherent propensity for occupational exposure or environmental contamination, and the ultimate disposal of the chemical must consider all the elements that I have mentioned earlier in the environmental fate or the exposure summary of the drug. Under probable routes of human exposures, we have an abstract from a survey which was conducted between 1981 and 1983. It has statistically estimated that 1358 workers are potentially exposed to 1-docosanol in the US. Occupational exposure to 1-docosanol may occur through dermal contact with this compound at workplaces where 1-docosanol is produced or used. Monitoring data for 1-docosanol, it says that the general population may be exposed via inhalation of ambient air or ingestion of food and drinking water or dermal contact with the compound or any other product that contains 1-docosanol. Now, although the number and the risk is huge, but still we have a proved safety profile for N-docosanol, which says that the dermal penetration of N-docosanol, it is minimum and it is just confined to dermis and stratum corneum, and systemic circulation bioavailability is also poor. So even though the people are exposed, but the risk of toxicity is very less. Now, FDA have approved N-docosanol under sections 505 of the Federal Food, Drug and Cosmetic Act on the basis of safety and effectiveness. It is also listed in the approved drug products with therapeutic equivalence evaluations list. The link for the information that I've used here is at the bottom of the slide. So this is the result of the clinical trial that was conducted for docosanol 10% cream topical to establish its effectiveness and hence its approval by the FDA. So this trial, it was 2 clinic initiated, double-blinded, placebo-controlled multi-center trial, and the patients, they were randomized to either docosanol 10% cream or placebo. The cream, it was applied before the beginning of an outbreak and before the development of a lesion 5 times daily. Total healing, it occurred when there was no evidence of an active lesion. These studies, they showed that the average healing time of a recurrent oral facial herpes simplex episode in 370 docosanol 10% cream treated patients was approximately 1 day shorter than they observed in the 367 placebo-treated patients. Hence, the claim on the label that Docosanel fastly heals the episodic lesions. In a subgroup in the same trial, it was noted that the Docosanel stopped cold sore episodes from progressing to the blistering stage in 34% of patients who applied it at the early signs of an outbreak, that is redness, as compared to the 23% in equivalent placebo group. Hence, the protocol that used Docosanel for best results at the very early signs of the infections, which were given out as tingling or pruritus or redness or a presence of bump. The side effects which was observed in the trial, it was headache, but then it was with the same frequency in patients treated with placebo as well, so it cannot be attributed purely to Docosanel use. So these are some studies that were carried out to establish the drug warnings for Docosanel 10% topical cream. These are published in 2006. It means it is post-approval studies, and the link for each one of them is given with the respective heading. The first one is the adverse events occurring in 2% or more of the patients receiving Docosanel. It included headache, application site reactions, and herpes simplex outside the treatment area, but the incidence is similar to placebo. The second is Docosanel 10% cream is recommended only for use on cold sores and fever blisters. So this study, it ruled out the possibility that Docosanel 10% can be used in genital herpes also. In the last study, it was a well-controlled study in pregnant women, and it does not show increased risk of fetal abnormalities despite adverse findings in animals or in the absence of adequate human studies. Animal studies, they showed no fetal risks. The chance of fetal harm, it is remote but remains a possibility. Although this is mentioned throughout the internet, but the information on the drug Docosanel, it does not include any specific FDA approved pregnancy data. The following costs are 2001 average wholesale pricing for the 3 creams that are available in the market for same indication. As you can see it on the screen, that Docosanel 10% cream, which is available as a 2-gram pack, it is much more cheaper than the rest of the 2 available dosage forms. At this point, please note that this is the only information that I could get on pricing of Docosanel as compared to the other available dosage forms or its competitors. Even Google itself, it was unable to provide me much information about pharmacoeconomic studies of Docosanel 10% cream. Under drug safety, we have mere repetition of the facts about Docosanel. Its systemic absorption is very limited, therefore the adverse events associated are strictly local. Dermatological reactions may include application site reactions or rash or dry skin or burning and stinging sensations. The more common side effect associated is headache, but still it is also with the same frequency in a placebo group in the clinical trials. FDA have classified Docosanel as pregnancy category B, but this information is still doubtful, as many of the sites displaying the monograph of Docosanel, they don't mention it there. So this is a newspaper announcement by Avani Pharmaceuticals. They issued it on May 29, 2000 after the approval and marketing of Docosanol. They have announced that the Canadian Therapeutic Products Directorate has issued a notice of compliance for Docosanol 10% cream, Abreva, the brand name, as a topical treatment for cold sores. And GlaxoSmithKline will be marketing the drug in Canadian market under an exclusive license agreement that provides royalties to Avanir based on wholesale product sales. So this is the continuation of the announcement, and it says that Abreva has grown to over 38% market share of the OTC lip remedies category in retail sales in US. It is the only cold sore product approved by the FDA for the OTC market and was recently recognized as #1 pharmacist's recommended cold sore treatment. The next para is about a statistic that the company provided or estimated about Canadian population and their suffering from HSV. They estimate that approximately 30% or roughly 7.5 million people in Canada will have outbreaks of at least one cold sore episode per year. Now, GlaxoSmithKline will be marketing Abreva in Canada as part of a license agreement signed by the two companies in March 2000. So this gives a right to the company to market the drug in both the United States and Canada. In Canada, however, provincial approval of the product is necessary before launch, so that is why the GlaxoSmithKline would be scheduling a presentation for National Drug Scheduling Recommendation Committee in September 2002. I might remind you that this announcement is May 29, 2002, so at that time it makes sense that the meeting would be held in September 2002. Now the drug is successfully marketed in Canada. Patent profile for Docosanol. The drug was meant to go off patent in 2009, but Avanir Pharmaceutical has had term of its key extended by 5 years to 2014 by the US Patent and Trademark Office. The extension, it triggered a $3.6 million payment from the Drug Royalty USA in accordance with the license purchase deal agreed between the two companies. According to this deal, Avanir received an initial payment of $20.5 million on December 24, 2002. Under the terms of the license purchase agreement with the drug royalty, Avanir retains rights to 50% of the royalties earned on sales of Abreva in excess of $62 million a year in North America, and retains the rights to all the current and future royalties from product sales in the rest of the world. Avanir, it also retains the rights to other potential product indications such as genital herpes and herpes zoster, but still these uses or these indications are not approved yet. Not long after the launch of Abreva into the market, both the companies, that is Avanir Pharmaceuticals and GlaxoSmithKline, had to face a lawsuit in which the plaintiffs alleged that Avanir and GSK have falsely advertised that Abreva cuts the healing time for cold sores in half. Now this case, it got dragged over 5 years, and in 2006 the plaintiffs were redefined as people who sued on behalf of themselves and all the other persons or entities in the United States who purchased Abreva from July 2001 through February 3, July 2006. On 15th March 2007, a Californian appeal court upheld a nationwide statement of false advertising claims about a cold sore medication, approving distribution of discount vouchers and a $1 million payment for medical research as compensation. Under the settlement approved by the trial court, the $1 million money it must be used to fund research that will benefit those suffering from cold sores, and it must not be related to any other current products except for vaccines, and it must be significant enough to be published in a peer-reviewed scientific journal. For consumers, individuals benefit, 50 million vouchers will be distributed in Sunday newspapers and will include a $3 discount coupon for Abreva. The trial, it also upheld attorneys' fees of up to $1.2 million. So this is a study which was published in June 2009, the same year when Dococinell was supposed to go off patent. In this study, the researchers, they tried to support the idea that Dococinell does not reduce the healing time with scientific proofs. So they said that Dococinell cream is barely no more effective than an excipient in treating acute episodes of herpes labialis or oral herpes simplex. The lifestyle measures are still the cornerstone of herpes management. Well, I think that it was not necessary, because after 2007 settlement with the court, both the companies, they left the idea of the fast healing, and they are not even mentioning it on the labels anymore. The reason that Dococinell got its patent extended and is still popular despite of the many negatives are these customer reviews. The consumer is happy about it, so it makes everybody else in the development process happy. So and at this point, if the ultimate user is happy and he is getting what he is bargained for, then the scientific proofs or the scientific literature, it doesn't matter if it's proving otherwise. Some of the customer reviews that I've mentioned here are fast healing, or blisters be gone, or best thing around for cold sores, etc. The review address is given in the red at the bottom of the slide. In the literature support for Dococinell, I have listed all the research papers and research which was conducted on Dococinell between 1980 to 2012. The underlined links are the respective addresses of each of the following research papers. The first one in 1980, as you can see it on the screen, it is a surfactant study, and Dococinell was treated merely a chemical compound. However, in 1982, when it was extracted from the bark of Pygium africanum, a whole new event of studies were designed to evaluate its uses in the human population. In 1989, the third study was established in which Dococinell was studied as a cellular marker compound in the identification of Mycobacterium xenopi. From 1991 to 1994, Dococinell was being studied as a broad-spectrum inhibitor against many lipid-enveloped viruses. In 1996, 3 studies were published. The main focus was to determine its antiviral activity against herpes simplex virus. In these studies, the important was double-blind placebo-controlled trial that led to its approval by the FDA. In 1998, the mechanism of action of N-docosanol against herpes simplex virus was studied and established. In 1999, the Progress in Research for New Drugs Against the Herpes Virus listed docosanol as one of them. In 2000, docosanol's new uses were explored, such as it reduced severity of phenol burn wounds in mice, and a review was published about its use in recurrent herpes labialis patients. In 2001, its use in type 1 HIV patients who are suffering from Kaposi's sarcoma lesions, they were studied for its effectiveness. In 2001, different review articles were published about docosanol and its relation with herpes simplex labialis and its effectiveness in the disease. In 2012, a toxicity study was published in relation to reproductive studies in rats and rabbits. These studies were conducted to establish its efficacy in case of genital herpes. These are usually called as phase 4 studies or post-marketing studies. By the end of 2002, genotoxicity studies were advanced and it included dogs as well. Some other review articles about docosanol's use for herpes labialis were published in the same year. In 2004, the synergistic inhibition of herpes simplex virus replication by docosanol and antiviral nucleoside analogues was studied. In 2006, advanced research for herpes simplex virus in hairless mice was studied. From 2006 till 2012, different review articles were published about docosanol and its effectiveness for herpes labialis. The last one in 2012 was the comparison of docosanol with other OTC drug and untreated controls. Here are the different references that I've used in my presentation. The most important and most yielding of all was PubChem, the second reference. It almost gave me all of the information that I have put in this presentation. Final words about docosanol, my presentation and experience so far. Docosanol is a miraculous medicine and it is proving to be very effective so far. Despite the many odds and negatives, it is still there in the market. It is still used by the majority because it is giving the consumers the results that they desire or they bargain for or they pay for. So if they are happy, the noble purpose of the drug is served. Throughout this course, we studied that the most important entity to be pleased or convinced in the drug development is the patient himself. So luckily docosanol is accepted publicly. Now I know that this presentation is lacking in many aspects, but trust me, it is the only information I would get my hands on after a search of over a week on internet. No matter how much or how many different words I would type on Google search engine, the result would be the same information. There was a lot of repetition and one way or the other, the links would connect to the same information. The site that did not help me at all was unfortunately the FDA site. It didn't even help me in a single word. I know, ironic, isn't it? Well, there was no IND package details, there was no NDA details, there wasn't anything about the pharmacoeconomic trials for docosanol. So that's pretty much everything that I have got here. Well, my efforts are in front of everyone. I hope it is appreciated. Thank you very much for your patience. Looking forward. Goodbye and have a nice day.